Neuroglia, Wikipedia audio. Neuroglia, also called glial cells or simply glia, are non-neuronal cells that maintain homeostasis, form myelin, and provide support and protection for neurons in the central and peripheral nervous systems. In the central nervous system, glial cells include oligodendrocytes, astrocytes, ependymal cells, and microglia, and in the peripheral nervous system glial cells include Schwann cells and satellite cells. The term derives from Greek gamma lambda alpha and gamma lambda omicron alpha GLU, pronounced in English as either /li/ or /la/. As the Greek name implies, glia are commonly known as the glue of the nervous system. However, this is not fully accurate. Neuroscience currently identifies four main functions of glial cells. Glia were discovered in 1856, by the pathologist Rudolf Virchow in his search for a connective tissue in the brain. For over a century, it was believed that the neuroglia did not play any role in neurotransmission. However, 21st century neuroscience has recognized that glial cells do have some effects on certain physiological processes like breathing, and in assisting the neurons to form synaptic connections between each other. Types: derived from ectodermal tissue. The most abundant type of macroglial cell in the CNS, astrocytes, have numerous projections that link neurons to their blood supply while forming the blood-brain barrier. They regulate the external chemical environment of neurons by removing excess potassium ions, and recycling neurotransmitters released during synaptic transmission. Astrocytes may regulate vasoconstriction and vasodilation by producing substances such as arachidonic acid, whose metabolites are vasoactive. Astrocytes signal each other using ADP. The gap junctions between astrocytes allow the messenger molecule IP3 to diffuse from one astrocyte to another. IP3 activates calcium channels on cellular organelles, releasing calcium into the cytoplasm. This calcium may stimulate the production of more IP3 and cause release of ADP through channels in the membrane made of pannexins. The net effect is a calcium wave that propagates from cell to cell. Extracellular release of ADP, and consequent activation of purinergic receptors on other astrocytes, may also mediate calcium waves in some cases. In general, there are two types of astrocytes, protoplasmic and fibrous, similar in function but distinct in morphology and distribution. Protoplasmic astrocytes have short, thick, highly branched processes and are typically found in gray matter. Fibrous astrocytes have long, thin, less branched processes and are more commonly found in white matter. It has recently been shown that astrocyte activity is linked to blood flow in the brain, and that this is what is actually being measured in fMRI. They also have been involved in neuronal circuits playing an inhibitory role after sensing changes in extracellular calcium. Oligodendrocytes are cells that coat axons in the central nervous system with their cell membrane, forming a specialized membrane differentiation called myelin, producing the myelin sheath. The myelin sheath provides insulation to the axon that allows electrical signals to propagate more efficiently. Ependymal cells, also named ependymocytes, line the spinal cord and the ventricular system of the brain. These cells are involved in the creation and secretion of cerebrospinal fluid and beat their cilia to help circulate the CSF and make up the blood-CSF barrier. They are also thought to act as neural stem cells. Radial glia cells arise from neuroepithelial cells after the onset of neurogenesis. Their differentiation abilities are more restricted than those of neuroepithelial cells. In the developing nervous system, radial glia function both as neuronal progenitors and as a scaffold upon which newborn neurons migrate. In the mature brain, the cerebellum and retina retain characteristic radial glial cells. In the cerebellum, these are Bergmann glia, which regulate synaptic plasticity. In the retina, the radial Muller cell is the glial cell that spans the thickness of the retina and, in addition to astroglial cells, participates in a bidirectional communication with neurons. Similar in function to oligodendrocytes, Schwann cells provide myelination to axons in the peripheral nervous system. They also have phagocytotic activity and clear cellular debris that allows for regrowth of PN's neurons. Macroglia. Satellite glial cells are small cells that surround neurons in sensory, sympathetic, and parasympathetic ganglia. These cells help regulate the external chemical environment. Like astrocytes, they are interconnected by gap junctions and respond to ADP by elevating intracellular concentration of calcium ions. They are highly sensitive to injury and inflammation, and appear to contribute to pathological states, such as chronic pain. Are found in the intrinsic ganglia of the digestive system. They are thought to have many roles in the enteric system, some related to homeostasis and muscular digestive processes. Microglia are specialized macrophages capable of phagocytosis that protect neurons of the central nervous system. They are derived from the earliest wave of mononuclear cells that originate in yolk sac blood islands early in development, and colonize the brain shortly after the neural precursors begin to differentiate. These cells are found in all regions of the brain and spinal cord. Microglial cells are small relative to macroglial cells, with changing shapes and oblong nuclei. They are mobile within the brain and multiply when the brain is damaged. In the healthy central nervous system, microglia processes constantly sample all aspects of their environment. In a healthy brain, microglia direct the immune response to brain damage and play an important role in the inflammation that accompanies the damage. Many diseases and disorders are associated with deficient microglia, such as Alzheimer's disease, Parkinson's disease, and ALS. Pituocytes from the posterior pituitary are glia cells with characteristics in common to astrocytes. Tannocytes in the median eminence of the hypothalamus are a type of ependymal cell that descend from radial glia and line the base of the third ventricle. In general, neuroglial cells are smaller than neurons. There are about 86 billion neurons and 85 billion non-neuronal cells in the human male brain. Glial cells make up about half the total volume of the brain and spinal cord. The ratio varies from one part of the brain to another. The glia/neuron ratio in the cerebral cortex is 3.72, 16.34 billion neurons, while that of the cerebellum is only 0.23. The ratio in the cerebral cortex gray matter is 1.48, with 3.76 for the gray and white matter combined. The ratio of the basal ganglia, diencephalon, and brainstem combined is 11.35. Most cerebral cortex glia are oligodendrocytes. Astrocytes account for 17.3% and microglia for 6.5%. Microglia. Other. Most glia are derived from ectodermal tissue of the developing embryo, in particular the neural tube and crest. The exception is microglia, which are derived from hematopoietic stem cells. In the adult, Microglia are largely a self-renewing population and are distinct from macrophages and monocytes, which infiltrate the injured and diseased CNS. Total number. Development. Capacity to divide. Functions. Neuron repair and development. In the central nervous system, glia develop from the ventricular zone of the neural tube. These glia include the oligodendrocytes, ependymal cells, and astrocytes. In the peripheral nervous system, glia derive from the neural crest. These pons glia include Schwann cells in nerves and satellite glial cells in ganglia. Current research involving glial cells in the human cochlea proposes that these cells are the common precursor to both mature Schwann cells and satellite glial cells. Additionally, the peripheral glial cells located along the peripheral processes expressed NGFR, indicating a phenotype distinct from the peripheral glial cells located along the central processes. Glia retain the ability to undergo cell division in adulthood, whereas most neurons cannot. The view is based on the general deficiency of the mature nervous system in replacing neurons after an injury, such as a stroke or trauma, while very often there is a profound proliferation of glia, or gliosis near or at the site of damage. However, detailed studies found no evidence that mature glia, such as astrocytes or oligodendrocytes, retain the ability of mitosis. Only the resident oligodendrocyte precursor cells seem to keep this ability after the nervous system matures. On the other hand, there are a few regions in the mature nervous system, such as the dentate gyrus of the hippocampus and the subventricular zone, where generation of new neurons can be observed. Myelin sheath creation. Glial cells are known to be capable of mitosis. By contrast, scientific understanding of whether neurons are permanently postmitotic, or capable of mitosis, is still developing. In the past, glia had been considered to lack certain features of neurons. For example, glial cells were not believed to have chemical synapses or to release transmitters. They were considered to be the passive bystanders of neural transmission. However, recent studies have shown this to be untrue. Some glial cells function primarily as the physical support for neurons. Others regulate the internal environment of the brain, especially the fluid surrounding neurons and their synapses, and neutrify neurons. During early embryogenesis, glial cells direct the migration of neurons and produce molecules that modify the growth of axons and dendrites. Glia are also crucial in the development of the nervous system and in processes such as synaptic plasticity and synaptogenesis. Glia have a role in the regulation of repair of neurons after injury. In the central nervous system, glia suppress repair. Glial cells known as astrocytes enlarge and proliferate to form a scar and produce inhibitory molecules that inhibit regrowth of a damaged or severed axon. In the peripheral nervous system, glial cells known as Schwann cells promote repair. After axonal injury, Schwann cells regress to an earlier developmental state to encourage regrowth of the axon. This difference between the CNS and the PNs raises hopes for the regeneration of nervous tissue in the CNS. For example, a spinal cord may be able to be repaired following injury or severance. Schwann cells are also known as neurilemocytes. These cells envelop nerve fibers of the PNs by winding repeatedly around a nerve fiber with the nucleus inside of it. This process creates a myelin sheath, which not only aids in conductivity but also assists in the regeneration of damaged fibers. Oligodendrocytes are another type of glial cell of the CNS. These dendrocytes resemble an octopus bulbous body and contain up to 15 arm-like processes. Each arm reaches out to a nerve fiber and spirals around it, creating a myelin sheath. This myelin sheath insulates the nerve fiber from the extracellular fluid as well as speeds up the signal conduction in the nerve fiber. Recent research indicates that glial cells of the hippocampus and cerebellum participate in synaptic transmission, regulate the clearance of neurotransmitters from the synaptic cleft, and release gliotransmitters such as ADP, which modulate synaptic function. Astrocytes are crucial in clearance of neurotransmitters from within the synaptic cleft, which provides distinction between arrival of action potentials and prevents toxic buildup of certain neurotransmitters such as glutamate. It is also thought that glia play a role in many neurological diseases, including Alzheimer's disease. Furthermore, at least in vitro, astrocytes can release gliotransmitter glutamate in response to certain stimulation. Another unique type of glial cell, the oligodendrocyte precursor cells or OPCs, have very well-defined and functional synapses from at least two major groups of neurons. The only notable differences between neurons and glial cells are neurons' possession of axons and dendrites, and capacity to generate action potentials. While glial cells in the PNs frequently assist in regeneration of lost neural functioning, loss of neurons in the CNS does not result in a similar reaction from neuroglia. In the CNS, regrowth will only happen if the trauma was mild, and not severe. When severe trauma presents itself, the survival of the remaining neurons becomes the optimal solution. However, some studies investigating the role of glial cells in Alzheimer's disease are beginning to contradict the usefulness of this feature, and even claim it can exacerbate the disease. In addition to impacting the potential repair of neurons in Alzheimer's disease, scarring and inflammation from glial cells have been further implicated in the degeneration of neurons caused by amyotrophic lateral sclerosis. Neurotransmission. In addition to neurodegenerative diseases, a wide range of harmful exposure, such as hypoxia, or physical trauma, can lead to the end result of physical damage to the CNS. Generally, when damage occurs to the CNS, glial cells cause apoptosis among the surrounding cellular bodies. Then, there is a large amount of microglial activity, which results in inflammation, and finally, there is a heavy release of growth-inhibiting molecules. Glia were first described in 1856 by the pathologist Rudolf Virchow in a comment to his 1846 publication on connective tissue. A more detailed description of glial cells was provided in the 1858 book Cellular Pathology by the same author. Clinical significance. When markers for different types of cells were analyzed, Einstein's brain was discovered to contain significantly more glia than normal brains in the left angular gyrus, an area thought to be responsible for mathematical processing and language. The ratio of glia to neurons increases with our definition of intelligence. Not only does the ratio of glia to neurons increase through evolution, but so does the size of the glia. Astroglial cells in the human have a volume 27 times greater than the same cells in the mouse's brain. History. These important scientific findings may begin to shift the neuron-specific perspective into a more holistic view of the brain which encompasses the glial cells as well. The glia's importance is becoming ever more clear as time goes on and new research is conducted. For the vast part of the last century, scientists have written off glial cells to be nothing more than the structure and foundations that hold the neurons in place. But now, there is direct evidence that correlates the number of glial cells in the brain with the amount of intelligence that any given species possesses. Future research will begin to shed light on the mysterious, yet increasingly crucial, role of glial cells. Bibliography.